Welcome back to a new video sessions of enzymology from Biology Boost Up. And today in this video we're going to learn about the classifications of enzymes and the action mechanism of enzymes. So now let's start. The classifications and nomenclature of enzymes. The modern system of enzyme classifications was introduced by International Union of Biochemistry in 1961. It groups the enzymes into following 6 categories. The first one is Oxido Reductase, and this type of enzyme take part in oxidations and reductions or transfer of electrons of a substrate. Basically, there are 3 types. First one is oxidase, second dehydrogenase, and third reductase. And the example of oxidoreductase is cytochrome oxidase. The second category of enzymes is transferase. They transfer a specific group from one molecule to another. As you can see here, during the synthesis of alanine, the amino group from glutamate transfer to pyruvate by the help of the transferase enzyme. The third category of enzyme is hydrolase. Those are enzymes which enable covalent bond to be broken by introducing of water. For example of hydrolase is protease, lipase, etc. As you can see, the A and B, two different kind of molecules attached by a single bond, and due to presence of hydrolase and H2O, those two compound totally separated and the bond totally break down. The fourth kind of enzyme is lyase. This enzyme causes cleavage or removal of groups without hydrolysis. Additions of group to double bonds or removal of groups producing double bonds. For example of lyase is histidine decarboxylase. Here two carbon atoms attached by a single bond, and A and B are two different type of molecules attached with each carbon by a single bond. And due to presence of lyase, those separate A and B molecule totally separated from the carbon. As a result, there are double bond has been formed between two carbon atoms. The next type of enzyme is isomerase. Isomerase cause rearrangement of molecule or structures of substrate. There are 3 types: isomerase, epimerase, and mutase. Now the last category of enzyme is ligase. Ligase catalyze bonding of two chemicals with the help of energy which is obtained from ATP, resulting in formation of such bonds as carbon-oxygen bond, carbon-sulfur bond, or carbon-nitrogen bonds. For example of ligase is pyruvate carboxylase. Now let's talk about the mechanism of enzyme action. The prime necessary for enzyme catalysis is that the substrate must combine with the enzyme at the active site to form enzyme substrate complex, which ultimately results in the product formation. This complex formation is a transient phenomenon during the substrate, where substrate is bound to the enzyme active site, a new structure is formed. Very soon after the expected bond breaking or making is completed, the products released from the active site. In other words, the structure of substrate gets transformed into the structure of product. Here E represent the enzyme and S represent the substrate. Both enzymes and substrate at first combined with each other and form enzyme substrate complex. Very soon after the formations of this complex, the products released from this complex. There are two different kind of model regarding the action mechanism of enzyme. First one is lock and key model and second one is induce fit model. So now let's talk about the lock and key model. This is also known as template hypothesis and which is proposed in 1894. According to this model, the structure of the enzyme is rigid. The substrate fits to the binding site just as a key fits into the particular lock or a hand into proper gloves. Thus, the active site of an enzyme is a rigid and preset template where only a specific substrate can bind. As you can see here, the structure of active site of an enzyme totally similar with the structure of a substrate, and that's why the enzyme substrate complex is formed easily. But on the other hand, according to the induce fit model, the active site of enzymes are not rigid. Rather, the enzyme change its shape upon binding substrate. The active site has a shape complementary to that of substrate only after the substrate is bound. As you can see here, the structure of active site of enzyme and the structure of substrate totally different, but still they form the enzyme substrate complex because the enzyme itself change its active site shape according to the substrate shape, and that's why this enzyme substrate complex is formed.